Hey guys, we are looking at the start here now of chapter 14, which deals kind of with similar things to chapter 13 in that we're still going to be talking about nucleic acids and kind of the molecular side of inheritance. But here we're going to switch gears a little bit and look at exactly how we get from genes that are passed from parents to offspring onto an actual phenotype, right? So we're going to look at here in chapter 14, how do we go from DNA, which is that genetic information we obtain from parents, and convert that into a phenotype with really in this case proteins, right? Remember, we've always said proteins are the things that are doing all of the activities and functions that we need in a cell. And so when we think about phenotype, that phenotype is derived from the proteins that are made from the genes we inherit in our DNA, okay? So that being said, this is a bit of a longer chapter as compared to chapter 13, and this first section is actually a little bit longer as well. So kind of bear with me as we go through this, but as always, make sure you've got your heading there, chapter 14, RNA and protein synthesis. And we're going to start with section 14.1 here, which deals specifically with RNA and a process that we call transcription, which is going to be something where we're going to start with our genes, our DNA, and we're going to talk about how we get from DNA into these molecules of RNA. And then as we get into 14.2, we'll talk about how we actually make proteins from those RNA molecules. But before we can do any of that, we have to figure out exactly what RNA is and compare that to what we've learned already about DNA in chapter 13. So our objectives are really just very short. I've only got two of them here. So we're looking at, again, those differences between what we're going to call RNA and DNA. And then we're going to talk about and look at how a cell makes different types of RNA. Okay. And as part of that, we're going to discuss that process that I mentioned, which is called transcription. All right. So when we think about RNA, and I went one too far there. RNA, unlike DNA, stands for ribonucleic acid. So DNA itself has, I'm going to scoot myself over here. DNA itself has deoxyribose, if you remember talking about that as part of chapter 13. That deoxyribose is the sugar that we find in the sugar phosphate backbone of DNA molecules. RNA in place of deoxyribose has ribose, which is still a sugar. The only difference is that we actually have an oxygen in a place where deoxyribose lacks an oxygen. But for all intents and purposes, the way we name this is very similar to how we name DNA. So the sugar in RNA is ribose. The other difference that we can easily see with these is that RNA here in this image is always single-stranded. There is no such thing as a double-stranded RNA molecule when we talk about our eukaryotic cells. We have double-stranded DNA, right? And we only have single-stranded RNA. And so in that case, that makes RNA a much simpler molecule. RNA generally is also only used as a temporary copy of something. It's not something that we maintain in our nucleus, similar to how we maintain our DNA. And so it's a little bit more fluid in terms of how it's produced and when it gets used. In addition to that, the other difference is that RNA has this other nitrogen-containing base called uracil, which is represented with the letter U. RNA does not have thymine, which is our T from chapter 13. Thymine is only present in DNA, and RNA is only present, uracil is only present in RNA, okay? So like we saw with thymine T, uracil is going to take its place and pair up with adenine in this case. So where we see an adenine in DNA, we would have a U or a uracil in the RNA that's complementary to that sequence. If we had an A in RNA, then we would still have a T if we had a complementary DNA molecule, right? Because we're comparing RNA and DNA. And we'll look at that a little bit here as we get into this process of transcription, okay? So going forward from that, looking at, again, kind of some of these differences summarized here. And I really like this chart. I feel like this is a super helpful way of kind of visually seeing all these differences. You can see again here, DNA, double-stranded, RNA, single, and still twists like we see with DNA. DNA here with our deoxyribose, and here's the position where we're lacking in oxygen. Here in ribose, it all looks the same except for this one tiny oxygen right here. All right, so again, same structure in terms of the sugar that we're looking at. RNA again has uracil, DNA has thymine, and then they all share in common cytosine, guanine, and adenine. Okay, so very similar in terms of what it's made up of. It's just we have these added parts now about ribose sugars and uracil in place of thymine. So there are 3 different types of RNA polymers. We have what we call messenger RNA, which is this right here. We have ribosomal RNA, which is represented with this image right here. And we have transfer RNA, which is represented right here. And each of these has a different function in a eukaryotic cell. And so we're going to go through each of these separately and individually. And I've got, I think, one slide for each. Yeah, one slide for each that we're going to talk about. So going first here into messenger RNA. I'm going to move myself around again. Messenger RNA is often just called mRNA, and that's usually the way that I'm going to refer to it in class also. mRNA molecules are, again, like I said, RNA tends to be more temporary anyway, and these are going to be very temporary in that they're used only for a short period of time. mRNA molecules are copies of specific genes, and they're produced so that we can go through this process of reading DNA, reading a gene, a code in DNA, and producing a protein. So messenger RNA is specifically kind of the product of this process that we're going to get to here in a few minutes, which is called transcription. The role of transcription in a cell, and we'll come back and talk about this again, is going to be really the first step in taking genes in DNA and starting to express them in the form of a protein. Okay, so mRNA is going to be that first major kind of connecting piece. It's the first thing we actually need when we want to go this route of making a protein. So we kind of start with DNA, we produce an mRNA molecule, and then that mRNA molecule, that copy of our gene, is going to end up going on and out of the nucleus to the ribosome, where it's going to be made into a protein. So mRNA, I always like to think of, is our connection between DNA and the genes we inherit and the proteins that we produce through this expression process. Okay, so in other words, connection between your genotype and your phenotype. So mRNA is pretty important, but it's called messenger RNA because that's its only job. It carries a message of a gene in DNA out of the nucleus and somewhere else. And the reason why we need it for that, it might seem like it's a little bit redundant. If we already have the sequence in DNA, right, why do we need to make a copy of that and then take it out? Why not just take out the DNA? Well, the danger in doing that would be that you could potentially damage that DNA, right? You could actually lose part of the gene or damage it in a way that it's irreparable. You can't fix it. So messenger RNA serves this role of kind of this temporary copy so that you don't damage the original, okay? And that's why we use it. So when we do transcription, this mRNA that we produce as a product of that ends up looking something like this. In that mRNA product, notice I've got several different things labeled. I have this thing called a 5' cap, a 5' UTR, which is an untranslated region. I have a coding region, which is what actually creates your protein later on, a 3' untranslated region, and then this poly A tail, which is essentially just a bunch of A's, a bunch of adenines all in a row. This gets modified, and we'll see that here in the very last slide of this section. All right, so we'll come back to what I'm referring to there at that point. The second type of RNA is called ribosomal RNA, and ribosomal RNA is important in a second process, which is called translation. So when we think about the production of a protein from a DNA molecule, we're going to transcribe it first, and then we're going to translate it second. And so as part of that transcription process, that's where we're going from DNA to mRNA. But now when we talk about translation, we have to go from mRNA to protein. And what helps with that process are these molecules that we call ribosomal RNA or rRNA. So rRNA has the important job of making up the ribosomes. Ribosomes, remember, are organelles that are not membrane bound. So they are essentially just ribosomal or rRNA interacting with different types of proteins to create these kind of unique structures, but they aren't like held within anything like a lot of the other organelles that we see. These are 2 images of different types of ribosomal RNA, and you can see, I mean, this is what it looks like when we model them using a computer. And then those same models kind of exposed and looking at them more deeply without all these twists and turns, you can see kind of how these chains are interacting with each other. So this is still truly a single-stranded RNA molecule, but what it does is it kind of folds up on itself and twists around based on little interactions that are happening between those single strands. Okay, but if you wanted to, you can go through here and choose one end of this thing and follow it all the way around. But they are complicated, again, but their major purpose is one thing, and that's to help make up the ribosome to help with this process called translation. Okay, so even though they might look complicated, don't worry about them so much. You just simply need to know what they're there for. Okay. The third type of RNA and the last type of RNA that you need to be familiar with is called transfer RNA, or also known as tRNA. And tRNAs work kind of in concert with rRNAs in that they both play a role in translation, but tRNAs also have this added complication in that they also have to be able to interact with mRNAs. So tRNAs are kind of our link. All right, they're going to be our connection here between mRNA and nucleic acid sequences and proteins. Because what we see forming here, and by the way, these are called tRNAs, they stand for transfer, but if you notice here, they actually kind of take on the shape of a T, which is helpful. But these tRNAs, again, single-stranded, right? They just fold over on top of each other and interact. At one end of this molecule, they have what we call an amino acid, and those are the monomers, if you remember, that make up our protein polymers. Glutamic acid here is one example of an amino acid monomer. And then at the other end, and all along this blue line, we have those nitrogen-containing bases, the nucleotides that make up our tRNA molecule. So I can see I have those nitrogen-containing bases here. I have 3 of them that stick out on one end, and that's going to be important for this translation process that we'll talk about as part of section 14.2, okay? But for right now, what I need you to understand is that tRNA takes on this shape. tRNA has parts of a protein here, but nucleic acid sequences down here. And tRNA stands for transfer. The reason why we call it transfer, by the way, is because it's transferring an amino acid, a monomer of a protein, over here to a growing chain of a protein, essentially, a protein polymer, okay? So that's going to be its role. So you can think of the T as representing transfer, you can think of it as representing translation, but either way, it's part of kind of both of those activities, okay? So this leads us into something called the central dogma, because I've been talking about transcription, I've been talking about translation, but I realize you know nothing about those, right? And so here's where I'm going to give you kind of that overview of those two processes and what all that has to do with protein expression, right? So the central dogma is like this central kind of unifying theory in biology. And this has everything to do with how we go from a genotype, right, genes that are inherited, to a phenotype or protein expression. And when we talk about the central dogma, this is always kind of routing us from. It's looking at how we take our DNA and how from that DNA we produce RNA that then is used to produce proteins. So if you look at this diagram here, I'm in the nucleus here, this purple area, and they're labeling, I've got a DNA strand. Now that DNA strand, when we unwind it, right, like we saw in replication, the machinery in the nucleus, the proteins present there would have access to a sequence of a gene, right? And so if a gene needs to be expressed because a cell is needing a specific protein, these machines, these little enzymes that we're going to talk about, go to that position in that chromosome. They know where to go. And in that position, they will actually start to unwind the double helix, and they start to create a complementary strand of mRNA. So here's my mRNA. That whole process, again, where they make that complementary copy of this gene is called transcription, and that happens here in the nucleus. And then if you see what's happening beyond that, that mRNA comes out of the nucleus through these nuclear pores and goes to find a ribosome. And that ribosome can be freely floating or it can be stuck on the rough ER. But either way, the process is going to be the same. The mRNA gets read by that ribosome, and we have this protein building process that happens as a consequence of that. So this is what we call translation right here. Okay, so we have transcription in the nucleus, translation at the ribosome. If we want to make this a little bit more simple, this is the diagram that I really like the most as we talk about this. And in looking at this kind of represents everything we've done in chapter 13 and 14 kind of all together. So central dogma, not only are we talking about going from DNA to mRNA to protein, we've also included now in this one that added process of replication. So when we think about, let me see if I don't. This blue arrow right here represents that, where we take DNA and we're going to make a copy of that DNA, right? This is DNA replication. What I would do if I were you is in your notes, I would have this diagram jotted down. And I would also add some things, some labels, so that you know what these arrows represent. So again, this blue arrow here, DNA to DNA, represents DNA replication. That was, again, chapter 13. If you remember from back in chapter 13, DNA replication is going to happen during synthesis, right? So this is also specific to the S stage. I mentioned that here because these next two processes where we produce RNA and then protein do not happen during synthesis. They happen when a cell is doing its normal cell functions. So though DNA replication happens during synthesis, DNA to RNA, or DNA to mRNA more specifically, which is, again, transcription, happens during G1 and or G2, okay? It can happen during either. So again, the red arrow here represents transcription. The green arrow here is going to be translation. And just like transcription, translation can happen, again, during G1 or G2. So really what that tells us is that transcription and translation are part of a cell's normal functions. The expression of a gene, the production of a protein, is going to be what a cell normally needs to do in order to maintain function, because again, proteins are what provide functions for cells, okay? So the production of this pathway, transcription and translation during G1, G2 is going to be part of that, okay? So I added down here at the bottom, and I like to think about it this way because I think it kind of puts things into perspective. RNA is a link between our DNA, our genes that we inherit in our chromosomes, what we know as our genotype, and the proteins that we produce, or what we know as our phenotype. Okay, so all the time we spent talking about inheritance and Punnett squares and pedigrees and Mendel's laws, that genotype is coming from the DNA, proteins are the phenotype, and RNA is the connecting piece between them. Okay. So if we focus here for a minute on transcription, so I'm going to focus here on this segment, DNA made into RNA. So again, that's happening in our nucleus, and it's happening during G1 and G2. Now, transcription is going to look an awful lot like DNA replication, which is great for you if you remember things about DNA replication. In some ways, it may be simpler. Hopefully, you think it's simpler. In the cell, it's the nucleus would certainly think it's also simpler, if only because we're not talking about copying all of the DNA in the nucleus. We're only talking about copying the code of one specific gene on one specific chromosome. So it's a whole lot faster, as long as those enzymes and those machines I mentioned on the last slide are able to go in and find that gene sequence. Okay. So when we talk about transcription, again, we're taking individual genes in DNA, and those genes are going to serve as templates. All right. So similar to what we saw in replication, a template is basically just kind of sitting still. It provides the code that we're going to copy. All right. And those templates are used to produce complementary mRNA. All right. So if we compare this to replication, couple of differences. In replication, we're making more DNA. We're copying the entire genome, all 46 chromosomes, and it's happening during synthesis. In transcription, we're making mRNA. We're copying only a single gene, and it happens during G1 and or G2, okay? Again, mRNA here, which you're seeing come out, this is going to be a single-stranded molecule, and you can see that represented here in this diagram too, okay? So what's going to happen as part of this process is we're going to use this transcription complex, which is essentially a whole group of proteins that work together to try and create a copy of this gene, okay? And this molecule right here called RNA polymerase is a big part of that. So if we look here, RNA polymerase is just a different way of representing it. This is actually from an old textbook, same general process though, okay? So I've still got my DNA here going left to right. I have my transcription complex here that would be comprised of RNA polymerase and several other types of genes. Sorry, not genes, excuse me, proteins and enzymes. And one of those other enzymes is a helicase enzyme. So if you remember in chapter 13 what helicase does, it's named because it helps to unwind the helix, right? Helicase, it breaks down the helix. So the helicase enzyme is going to be part of this transcription complex, this big giant yellow blob you see right here. And that transcription complex is going to find the start of a gene. Once it finds the start of a gene, or the gene of interest, right, whatever gene it's trying to make a copy of, we're going to start to unwind the double helix at that position and only at that position. So we're not unwinding the whole chromosome, right? We're just focused on one specific area. So helicase comes in, it starts to unwind the double helix, and it unwinds maybe 10 to 20 nucleotides at a time, okay? And as that happens, we have other enzymes that are going to start to become active, and they play their specific jobs. So you can see here, I have free-floating nucleotides in the nucleus, just like we saw in replication, except again here, they're going to have ribose sugars, and they're going to have uracil, not thymine. Okay, but just like in replication, these free-floating nucleotides come in, they interact with their complementary base pairs, but again, only on one side of this DNA template. Okay, again, we're only making one copy of this mRNA. So as they come in and start to interact, right, we form a new molecule of mRNA. So this red backbone color here is my new single-stranded RNA. And notice I'm not doing anything with this top part of DNA, right? This upper strand. I'm not copying that at all. So we're using the bottom strand here as our template, and we're making that mRNA. Similar again to replication, we also have a polymerase enzyme that's part of this process. In this case, we call it RNA polymerase because its job is to form the backbone in RNA molecules. So it's going to form covalent bonds between the sugars and phosphates that make up the backbone of the new mRNA. Okay, RNA polymerase does not function in replication, just like DNA polymerase does not function here in transcription. So these polymerase enzymes are specific for the types of nucleotides that they interact with. Okay. As RNA polymerase does that, this transcription complex will continue to kind of move along that DNA molecule. And again, once we've got about 20 or so kind of transcribed and that mRNA is starting to be made, the RNA is going to start to kind of pull off and detach. The helix will start to rewind behind, that space that's been completed. And so really at any one time, we don't have a whole lot of DNA that's kind of exposed. It's very specific to which gene is being read and the position and the progress of that process, okay? So once all this is done, again, mRNA is going to detach. It'll be freely floating in the nucleus for a bit. And the very last thing that has to be done is a little bit of mRNA editing. And so when we talk about editing, what this means, remember I said a long time ago or a while back in the screencast, we had different parts of this RNA molecule, right? So in the different parts, I have these caps and tails. I have other little segments here. I mentioned they were UTRs, which is untranslated regions, things that don't need to be read by a ribosome. But I also have these other things called introns. And introns are called introns because they're interfering elements. What that means is that they interfere with the actual gene sequence. Why they're there, nobody really knows, but we do know that they have to be removed. We say that introns have to be spliced or cut out of that mRNA molecule, or what they're labeling here is pre-mRNA. So my introns actually here make up a good percentage of the molecule that was just created, and that's pretty typical. Okay, introns tend to make up a large portion of our chromosomes, even though they never get translated and they don't make anything in terms of our phenotype. They're just there and they're in the way. And so there are special enzymes in the nucleus that'll come along and chop this mRNA molecule at these positions to remove those lighter blue introns here. And then all that mRNA gets squashed together and it's ready to go on to the next stage, okay? So again, the next stage is going to be looking at exactly how translation occurs. So then How do we deal with this mRNA copy that's been made, and how do we actually get that into the form of a protein, okay? And for that, it does get a little bit more complicated and different. This one, again, is very similar to replication. I hope that you saw some of those similarities, all right? So we will keep practicing with all this, but as always, let me know if you've got any questions. Otherwise, I will see you next time for 14.2. Bye, guys.